UIEC. There we go. Okay.
All right. Good afternoon, everyone. My name is Charles Blatti. I'm a postdoc at the Knowing BD2K Center at U of I. Today, I'd like to talk to you a little bit about the analysis tools we develop at the center, and then specifically a project in which we collaborated with the Seven Bridges Cancer Genomics Cloud in order to bring our analysis tools into their compute ecosystem, and then finish up with a few example applications of using those imported tools in the Seven Bridges environment. So just a little bit about Knowing. So one of the focuses of our BD2K Center is that we are always trying to develop and use prior knowledge about genes and proteins in the analysis of omics spreadsheets. So researchers will do experimental assays to profile different samples and conditions. We think of that information in terms of an omics spreadsheet, where the samples are the columns of the spreadsheet, and then the genes, the transcripts, the proteins that were profiled make up the feature rows of the spreadsheet. And then there are many analysis techniques that people will want to do on these spreadsheets. Find clusters of samples that relate to genomic subtypes, find gene features that most correlate with the phenotype information you might have on the samples, or just find known gene expression signatures or gene sets that relate to the samples individually. And so you can do these analyses with the omics spreadsheet alone, but at Knowing, we think, well, the features are all gene features, and there's a lot of other information out there, prior knowledge we have on relationships between genes and the annotations of genes. And so it didn't show up great, but on the right you see a network where there are edges in between genes representing interactions between those entities, and then also edges between genes and properties for known annotations. And because the prior knowledge genes relate to the features of our omics spreadsheet, what we'd like to do is the analysis of the spreadsheet while also mining the graph for the relationships in the prior knowledge. And so there are many different types of prior knowledge on genes and proteins. You can have experimental relationships defined by co-expression or physical interactions. You can have curated annotations, pathway and gene ontology are examples. You can have computational relationships between the genes. Edges created from text mining co-occurrence, from shared protein domains, or from sequence similarity. And so at Knowing, we gather these different types of relationships on these gene-protein entities together from different databases, and we store them, and we call it the knowledge network. And we've applied, so we use this knowledge network to inform our analysis, and we've applied different analysis tools in many different domains. We've used protein interactions to help prioritize genes that correlate with the variance in drug response. We've used conservation networks in order to find conserved or consistent gene ontology annotations across multiple species in their social behavior gene expression sets. And then other research groups have shown that text mining based networks can help overcome the problem of sparsity in TCGA mutation data and find meaningful genomic subtypes in a clustering analysis. And so these different types of analysis algorithms, the Knowing Center, we make them available on our platform. The basic idea is that a researcher will come to us. They'll have their raw omics data on their laptop. Oh wow, there's supposed to be a cloud there. They will load their spreadsheet of omics data into our Knowing compute cloud, and once that's in there on the compute cloud, they'll select the analysis workflow that they're interested in, and then the knowledge network information that they'd like to include in that analysis, and then the results will be displayed back to them through our interface. And there are, if you can see, there's supposed to be two different clouds that'll be important for a slide in the future, which you also probably won't be able to see. So the Knowing Analysis platform is available now. If you would like to try it out, it's knowing.org/analyze. You can sign up. There's demo data sets in there that you can run any of the pipelines on, or you can prepare your data and run it on your own data sets. And we have many different types of analysis tools with both standard methods and knowledge guided versions available. And the current knowledge network that's available on the platform is very generalized. It supports 20 different species. It has nearly 500 million relationship edges in it, and of almost 200 different edge types. So the project, though, I'm planning to talk to you about is not about our platform, but about a different platform. And so this is the 7 Bridges Cancer Genomic compute platform, and we've had a very nice collaboration with them over the past year. And so 7 Bridges, unlike our platform, stores some of this raw, or has available this raw omics data for very specialized human disease data sets. And because their focus is on this specific set of data, they have also, and make available also, customizable query interfaces that really allow you to select and parse and filter the exact data files from these large public data sets that you're interested in. And the CGC, which I'll say for shorthand for probably the rest of the talk, also has very nice data access management features for controlled data sets, and has a payment system for the amount of compute cloud usage you're using. And so the main idea of the collaboration with the 7 Bridges was that we would like to bring the Knowing knowledge-guided analysis to this specialized platform. And we're trying to bring the tools where the big data resides. And so here's another picture. There are clouds there that you cannot see. But the idea in this one is that the user will now go to the CGC interface. They will use their tools to select and filter the TCGA data, for example, that they wish to analyze. They'll convert that into a spreadsheet, and then from the CGC, within the CGC compute cluster, they'll be able to run the Knowing analyses using the knowledge network data, and also then finally get the results back to them in the CGC interface. And there are 2 technologies that we use to make this project work. One of them is Docker, which just enables your tools to be run on any machine anywhere. So it brings the binaries, the libraries, and the dependencies along with the tool inside Docker containers. And so gene-set characterization is one of our analysis tools. First, we must containerize it to do this import into the CGC. And all of our containerized tools are publicly available on a Docker image repository where they're versioned and then can be shared. The second technology that we use in order to import the Knowing analysis tools into the 7 Bridges Cancer Genomics Cloud is the common workflow language. So this is a standardized way to describe the inputs, the outputs, the execution steps, and the requirements of your analysis tool and your workflow tasks. Once you describe your tool with a CWL description, it is more readily run by standardized tool executors. The CWL can actually be used to visualize inputs and outputs to your tool, or to create interfaces to help you modify the parameters. And then the CWL will, because you've specified the different parts of individual tools, also enables you to string together multiple tools together in multi-step workflows. And just a side note, so the CWL can be stored in sort of specialized repositories like the doc store, where they will link to your Docker image and have the tool description. And then because our tools were written with CWL descriptions and with Docker images, we were able to participate in the GAG4H Dream Workflow Execution Challenge. We had our gene prioritization tool in that challenge, and it was run on 11 different compute clouds successfully by the challenge participants. So back to the task of the main challenge of this project, which was to get the knowing tools available and running in the Seven Bridges Cancer Genomics cloud. What are some of the practical hurdles that we faced when doing this? Luckily, they weren't as large as they could have been because Seven Bridges has done a nice job to accelerate the process. They have a built-in common workflow language importer. So once your tool has a description, you can import that description directly into their platform. And then you might have to tweak it and test it to make sure it runs well. And so they have a SDK Rabix composer that they wrote, which gives you access to visualizations, the code, and the testing that you might need in order to make sure everything runs smoothly. And so using their importer and with their help, we've got 4 of the analysis tools that are on our platform, also on the Seven Bridges Cancer Genomics cloud. They're all publicly available. You can go to that URL if you want to give them a try. You can also find them on our knowing/analyze, links to them on our knowing/analyze website. One of the tools that we have public in the CGC, we had to build specific for use in the CGC. So it's not something that exists in our platform. So the CGC will have data files specific to their data sets, and what we needed was a tool that can take those data files, RNA-seq, FPKM files, for example, and convert them to a spreadsheet that the rest of our analysis tools then expect. Okay, and so the process was successful. It wasn't as painful because we've been using the technologies that enable interoperability. And so I wanted to spend the rest of it, my time, just giving you a flavor of some of the things that are now possible because knowing analysis is available in the CGC. So the first is just a small application where we're going to recreate the analysis from one of last year's TCGA papers entirely within the CGC. So the paper we chose was from the esophageal carcinoma paper. One of the analysis they ran was what we call signature similarity analysis. And in this particular instance, they had 90 esophageal squamous cell carcinomas, tumor samples, and then they wanted to understand and map those tumor samples to previously characterized signatures of lung squamous cell cancer. And so these lung squamous cell cancer data sets came from a previous paper, and they were basal, classical, primitive, and secretory. And so we are going to, I just want to walk you through the steps that are required in order to recreate the analysis from the TCGA paper. So first in the CGC, you'll use their specialized querying interfaces in order to select the 90 tumor samples that are of interest to this data set. The second step is to feed those 90 data files into the Knowing spreadsheet builder, which will convert them and format them for any other downstream analysis by Knowing analysis tools. And then finally, we just have to run the Knowing signature analysis pipeline and hit go and hit run. And so the image on the right is just a sample of what you might see in the CGC when you're setting up this process and running this process. The inputs are on the left. In the center are the different parameters that are specific to the tool. And then when the run is complete, you see the outputs that are generated. And so with about 20 button clicks, we can launch, that are necessary to launch this run. And if it takes the analysis tools to use the cloud compute resources for about 13 minutes, and that ends up costing us about 6 cents, we can recreate this particular part of this TCGA analysis. So the top panel is the figure from the paper. The bottom panel is from our rerun of this analysis. You can see that they're not quite identical. So the overall finding that they report in the paper is the same. So each subtype is, each of their clusters is mapped to the same primary subtype. And the reason for the difference is a lot of it has to do with how the RNA-seq data was quantified and normalized, which is different in the paper than on the data files on the CGC. But since we're talking about reproducibility, if you wanted to reproduce the bottom panel, it's really, really easy because you can share this run in the CGC with anyone and give them a copy, and they will have the same versioned data and the same version analysis tools, and they can rerun and they'll generate the exact same figure. They could also change the parameter or change the inputs. So the bottom panel represents, well, let's just redo the signature analysis, but instead of using the 90 ESCC samples, we'll use all 550 lung squamous cell carcinoma samples. And it's just as easy as a few button clicks to rerun the results on new data or with new parameters. And so that is recreating the analysis that exists in the paper. So the last little section I wanted to talk about how you might use this tool, the knowing tools in the CGC, to sort of extend beyond the analysis, running multi-step analysis, and also to highlight our knowledge-guided gene set characterization pipeline. So the task that we are going to assign ourselves is to try to characterize the genes that distinguish each of the genomic subtypes within these 90 ESCC samples. And in order to do that, we already have mapped them to their best matching signature. We're now going to run gene prioritization for each of those signature subtypes, and we'll find the top 100 differentially expressed genes between the samples that are most similar to that subtype and then all other samples. And so this will give us 4 gene lists, each with 100, each for each different genomic subtype. And what we're going to do is we're just going to do gene set characterization on those 4 gene lists. The standard method is the Fisher exact test, which is a statistical enrichment test, which you would probably be familiar with if you've ever used the tool David. And then we're also going to run, besides the standard method, a knowledge-guided gene set characterization method. So this is the drawer method developed in the center stands for discriminative random walk. And the basic idea is that it uses guilt by association techniques on a heterogeneous knowledge network. And so, for example, the first step in building the network, my blue nodes are the genes, my red squares are the different properties or pathways which we're going to try to relate to them. We have the annotations of the genes for the pathways are the red dotted lines, and then additional relationships we know between the genes are represented by the solid blue lines. And the basic idea of the drawer method is that you'll have a random walker. It will start at our differentially expressed genes, and it will probabilistically follow the edges of the graph or return to one of the start nodes. And it will repeat this process many times, and eventually it will create a distribution over all of the nodes that can be thought of as a proximity score of each node to the original restart set. And then finally, it will rank all of the property nodes in the network based on their relative proximity score over an appropriate baseline. And so we did this for all 4 subtypes, so 4 100 differential expression lists. We ran the Fisher method and the drawer method and looked at the enriched pathways. And I focused on just selecting the best 10 pathways per method. And this first table shows you the pathways in which both methods agreed. So you can see the colored columns show that both methods ranked that pathway particularly high for the subtype. The Fisher p-value column shows that the standard enrichment was very significant. And then the final column, many of these pathways, there's literature evidence that they are involved in esophageal squamous cell or other squamous cell studies. And many of them, strangely, are similar between the different subtypes. So the NRF2 is shared between basal and classical. Pathway is significant for both basal and classical. But then there are also several pathways that are specific to each subtype, which is sort of novel information that can be then brought to bear when treating patients with that particular subtype. And then the final table I have is now not the shared pathways, but the pathways that will only come out with the drawer analysis. And so this is, if you look at this, the rank for drawer is strong, the rank for Fisher is poor. The Fisher p-values of significance are low or high, not significant. And you still see nearly as much, or probably as more literature evidence that these are real terms that are already known to relate to ESCC. So this means that the genes in the pathways themselves are not differentially expressed, but perhaps the neighbors of the genes in the pathways are differentially expressed, which is why drawer is able to pull them out. And I just wanted to highlight one result. So this FOXM1 was recognized as a possible target for treatment of ESCC patients, and the fact that we found that the FOXM1 transcription factor network relates particularly well to one of the subtypes of ESCC would perhaps be, if they were going to follow through and develop it as a therapeutic, that would be something they would want to investigate to confirm if it has a relationship with that subtype. And so that was my talk. The high level is that we have the knowing knowledge analysis, and we've been able to bring it to the human disease data sets that exist in the CGC. You can recreate TCGA analysis. You can extend them with knowledge guided. I'd like to thank the funders and the people who invited me, Seven Bridges collaborators who've been great over the year, and then especially Milt Epstein and Amin Ahmad at Knowing, but all of the development team for most of this is somebody else's work. And there's a lot of Knowing talks, or a few Knowing talks coming up. So if you want to learn more about the algorithms, Saurabh is talking tomorrow. If you want to learn more about the platform, Colleen is talking on Monday. And Jin Feng is a graduate student who's developed a more advanced signature analysis tool, and he'll be also talking on Monday. Thank you. Any questions? Oh, so the Knowing Spreadsheet Builder, which is the one tool we had to build special for the CGC, what it does is in Seven Bridges, if you ask for the RNA-seq data from the TCGA, you get individual files with a column of FPKM numbers. And so all the Knowing tools were built assuming that you get this matrix where the samples have already been merged into one big spreadsheet rather than the samples each are different files. And so the Spreadsheet Builder is just handling that work of merging the different data files, making sure the genes map to the correct entities. It has some very basic normalization and filtering capabilities in case you know you want to discard some of the things that you would get in the raw CGC data file. Yes?
So how would you compare your solution against like AWS and Google when you also have like containerized environments and also
So I do not have any experience using the what is the AWS one called? So what Seven Bridges is actually doing in the back end is it's just taking our containers, it is spinning up AWS EC2 instances, and it is running our code on an AWS EC2 instances and then saving it in some S3 bucket so you can access it later. And so it is, I think, the advantage of going with something like the Seven Bridges is that they've done all the work to sort of curate the data sets, find quick ways to access the data you might want from the data sets, and there's a whole wealth of different genomics and bioinformatics tools that they also support in there that you can connect the downstream analysis with our sort of more upstream analysis all in one place without having an all-purpose container execution engine.
Any other questions? Okay, well, I'd like to thank all of the speakers once more, so can you give me a round of applause? Great talk.